Hello, everyone, and welcome back to Chapter 2, which is entitled How We See the Invisible World. In Chapter 1, we took a look at the 6 categories of microscopic organisms that inhabit this invisible world. And we also took a look at a brief history of microbiology, noting how important the microscope was for being able to develop this field. So, Chapter 2 is dedicated to looking at this tool, the microscope, that we use to peer into this invisible world, how it works, how we use it, and how we prepare samples and specimens for visualization under the microscope. If you look at this section in your textbook, you'll be able to see that there are many different types of microscopy out there. There's light microscopy, which, if you use the microscope for it, is most likely a light microscope. Light microscopy uses beams of visible light to illuminate a specimen and create a picture that reaches your naked eye in a magnified form. But that's not the only form of microscopy out there. There are also much more sophisticated and sensitive microscopes, such as scanning acoustic microscopy, which uses sound waves instead of light waves to create an image. Electron microscopy uses beams of electrons, and these beams of electrons are able to magnify a much greater degree and create much greater resolving power to look at smaller types of organisms such as viruses. Scanned probe microscopy inserts a tiny probe into a specimen and then transmits an electric current through the probe in order to map the structure of the specimen. However, for our purposes, we are going to be focusing only on light microscopy in this class and limiting our scope within light microscopy to just 4 of the different subtypes of light microscopy. And those are bright field microscopy, the type that you will be using in class, dark field microscopy, phase contrast microscopy, and fluorescence microscopy. Before we move on to take a look at each of these types of microscopy in greater detail, we're going to look at a standard microscope first and check out each of the different parts and the role that they play in the function of the microscope. The type of microscope that you see here on the slide is called a compound light microscope. Of course, it's called a light microscope because it uses light which comes from the illuminator or the light source located just here. And it's called a compound light microscope because it uses a compound, or in other words, a combination of 2 lenses in order to magnify a specimen. One of those lenses is called the ocular lens, sometimes referred to as the eyepiece. And this lens is the one that you look into. You place your eyes adjacent to this lens and look down at your specimen. There's another lens here called the objective lens, and the objective lens actually rotates and can be switched out for usually one of the 4 different magnifications that are available on the same microscope. The objective lenses are mounted to this piece right here, which is called the arm of the microscope. And the body tube is where the ocular lenses are mounted. The body tube is what transmits the light up from the light source through the objective lens, through the body tube, and then through the ocular lens, where it finally reaches your eye, and you get a magnified image of whatever is sitting here on your microscope stage, which is the black platform where you place the microscope slide. Along the way, the light will pass through a few other components of the microscope. We have the diaphragm and the condenser, which are mounted just below the microscope stage. And the diaphragm is sort of like a shutter on a camera. It has a lever and it allows you to control the amount of light that's entering the condenser. And the condenser is a special glass lens which will take the light coming up from the illuminator and condense it into a point which will hit your slide. On the side of the microscope, we have two knobs that are used to focus the microscope, which are mounted on the base. The coarse focusing knob is used to raise and lower the microscope stage, either closer to or farther away from these objective lenses. The fine focusing knob is used to bring the specimen into fine focus after the coarse focusing knob has allowed you to get an initial picture of what you're looking at. Here on the right hand side, we can see a simplified image of the microscope where the path that the light takes has been highlighted up from the illuminator through the condenser lenses, which turn that light into a pinpoint beam that hits the specimen, which then moves up into the objective lenses through the body tube, through a prism, and then it's reflected in your eyes through the ocular lens. Now, as I said, the microscopes typically have 4 different objective lenses that can be rotated out to get different degrees of magnification. Usually those objective lenses are the scanning lens, which is the lowest magnification. The magnification of this lens is 4x, meaning 4 times greater than the naked eye. Then there's the low power lens, which is 10x, meaning 10 times greater than the naked eye. The high power lens is 40x, and the oil immersion lens, which is 100x. And you can see those 4 listed right here in this image. And one other thing I want to mention is that the ocular lens also gives magnification, but this gives a set magnification. Unlike the objective lenses, which can be rotated and changed out for 4 different degrees of magnification, the ocular lens always provides the same magnification, and it is almost always a 10x magnification. Now, the names of these 4 lenses for the most part make sense to us intuitively. The scanning lens, which is the lowest power lens, is used to scan around in the field of vision on the slide and try to identify the specimen that you're looking for. The low power lens is named for the fact that it has lower power, high power because it has higher power, and then comes the oil immersion lens. The oil immersion lens requires a bit more explanation as to why it's called the oil immersion lens. The immersion oil lens is used in conjunction with a special substance called immersion oil. And the reason why this is the case has to do with the combination of something called the working distance and refractive index. As you saw on the previous slide, you may have noticed that the different objective lenses were all different lengths and sizes. For example, the 4x lens is very short and stubby. The 10x is longer. The 40x is longer still, and the longest lens of them all is the 100x objective lens. This means that when you are working with the microscope, the distance between the tip of your lens and the slide is different depending upon which objective lens you are set to. And this distance here is what we call the working distance. The oil immersion lens, being the longest of all the lenses, has the smallest working distance between the tip of the lens and the specimen on the slide. As it turns out, this has an impact on your ability to visualize the image on the slide because of a property of light called the refractive index. Refractive index describes how much a beam of light bends as it passes from one substance into another substance. Refractive index explains why, when you're looking at an interface between water and air, you get these distorted images like the one that you see here with this drinking straw, where the straw appears to be in one location, and then at the interface between the water and the air and the glass, it becomes distorted. That's because when the light passes from the water into the open air, the light bends. And that's because the speed at which the light passes through the water is different from the speed at which it passes through the air. This affects how you see this image here. Now, when it comes to the microscope, getting back to that special lens called the oil immersion lens, when a beam of light is traveling up from the light source, it will pass through your glass microscope slide, and then when that light passes from the slide out into the open air, that beam of light will bend because, again, the refractive index of glass is different from the refractive index of air. Because the oil immersion lens is so close to the microscope slide, and because there is such a small working distance here, a lot of that bent light is going to escape. Rather than traveling up into the objective lens and on the path toward your eyes, this refracted light is going to be essentially spread out, and it won't be captured by the objective lens. The solution to this problem is to add a drop of immersion oil to the surface of the glass slide. Compared to air, the refractive index of oil is more similar to the refractive index of glass. What this means is that when a beam of light passes up through the glass microscope slide and into oil rather than air, it will bend less when it passes from glass into open air. That means that the light is going to pass into the objective lens and travel up towards your eye rather than refracting outward and being lost. If this drop of immersion oil is not added when using that 100x objective lens, then typically what you'll get is a very dark image where you can't really discern anything about your specimen. In order to get full proper illumination, the immersion oil has to be added to increase the light capture due to the refractive index of light. Now, the total magnification that reaches your eyes is going to be a combination of the two lenses that are present in a compound light microscope. So there's two lenses, as we've just seen. There's the objective lens and the ocular lens. The ocular lens is almost always 10x. The objective lens is going to be the one of the four options we've just seen. The total magnification is the product of the two. The value of the objective lens multiplied by the value of the ocular lens. So this brings us to our first checkpoint. Checkpoint number 1. If the ocular lens power is 10x and the microscope is set to the high power objective lens, the magnification of which is 40x, what is going to be the total magnification of the specimen as you see it? Now that we've checked out the basic functioning of a light microscope, we're going to move on to talking about the 4 different subtypes of light microscopy that we are going to focus on in this class. The first and simplest is called bright field microscopy. This is the most common type of microscopy, which uses visible light to illuminate a specimen from behind, so the specimen appears dark against a bright background, as you can see here with this image of a paramecium. Bright field microscopy can be contrasted to its counterpart, which is dark field microscopy. In dark field microscopy, there is an additional component to the microscope, a special disc that sits above the light source between the slide and the condenser. This special disc prevents a large majority of the light from entering the objective lens. Only light from the specimen itself and not light from the surrounding slide will enter the objective lens. The result is that the specimen appears against a dark background instead of a light background. This image right here shows you the difference between a bright field microscopy setup and a dark field microscopy setup. As you can see, in a bright field microscope, the light passes through the condenser lens, where it is condensed to a pinpoint on the slide, and then it enters the objective lens. In a dark field microscopy setup, we have this special disc which blocks a majority of the light. The light that remains gets condensed just onto the specimen, and light from the surrounding area of the slide does not enter the objective lens. So as you can see, what we get is a bright specimen up against a dark background. And dark field microscopy is useful for special circumstances. Mostly it's when you need to take a look at live organisms or organisms that resist staining. Sometimes organisms must be preserved and kept alive when you're looking at them under the microscope. And when this is the case, dark field microscopy is very useful. Other organisms do not stain well with the special dyes that we use in bright field microscopy. And in this case, dark field can also be helpful because it creates this artificial contrast between the specimen and the background. Next, we have phase contrast microscopy. In phase contrast microscopy, two types of light end up entering the objective lens. There's the light directly coming from the illuminator, and then there's light that is transmitted through the specimen. Because there are two types of light, this creates a more complex image which adds contrast that allows you to visualize internal cell structures. And it also allows you to visualize live specimens and their structures as they are in motion. For example, when you're viewing a specimen in phase contrast microscopy, you do not need to stain it with any artificial dyes in order to visualize it. So here we have a picture that shows us a side-by-side comparison of these first three types of microscopy. We've got paramecium under bright field, paramecium under dark field, and a paramecium under phase contrast microscopy. The fourth and final type of microscopy that we're going to look at in this class is fluorescence microscopy. Fluorescence microscopy uses fluorochromes, which are fluorescent dyes, to stain organisms and visualize them in a fluorescent hue. In this case, the light source that comes up from the illuminator is ultraviolet or near ultraviolet in wavelength. Samples in fluorescence microscopy appear in a bright fluorescent color up against a black background. And fluorescence microscopy has special purposes. Certain species of bacteria can be effectively stained and detected with this technique, so it can be used to identify pathogens. For example, Treponema pallidum is the species of bacteria that causes the STD syphilis. And in this image, it has been fluorescently stained and dyed so that it's visualizable. They can also be used to distinguish live cells from dead cells. So here, for example, is an image of a fluorescent live dead assay that one of my colleagues took when she was working at ASU. And in this type of fluorescence microscopy, there are sections of these filamented cells that are still alive, stained green, and there are sections that are dead where the membrane has been breached, and those are stained in red. So these are a few purposes of fluorescence microscopy. This slide right here is a great summary slide from your textbook, which compares the bright field, dark field, phase contrast, and fluorescent microscopies very concisely on the same slide here. So here we have a checkpoint about the different types of microscopy. I want you to tell me which type of microscopy would be best for viewing the storage organelles called vacuoles inside of fungal cells, which is what you can see pointed out in this image right here with the arrows. So, which of the four types of microscopies would be best suited to this purpose? So, the next step in this lecture is to take a look at the two processes that we use to prepare samples for visualization when using light microscopy. Those processes are called smearing and staining. Staining is the process of coloring a specimen with a dye that is used to emphasize either the whole cell or certain structures. There are different types of staining which we'll go over momentarily, But before staining, a specimen, it must be fixed, or in other words, attached to a glass microscope slide in a thin layer called a smear. So in other words, a sample of microorganisms must be smeared on the surface of a glass microscope slide, and it must be fixed using heat to cause it to adhere and attach to the slide. And then color can be applied so that it can be visualized under the microscope. The process of staining and smearing is one that we will do in the lab, but to give you a quick overview of how this process works, essentially a sample of bacteria or other microscopic organism is transferred onto the surface of a microscope slide and spread in a small area between the size of a dime and a quarter. It is then allowed to completely and fully air dry. Then that smear is then passed through the heat of a Bunsen burner or allowed to heat in front of a baccinerator for a period of time. And by heating the dried out slide, this causes the microbial cells to become adhered to or glued to the surface of the microscope slide. Why is this important? Well, it's important because you're about to apply a stain or a dye, and when you go through the process of applying that stain, you want to make sure that the liquid does not cause the cells to wash off the surface of the slide. Hence, heat fixation, as it's called, is necessary to finalize your smear before you begin staining. Now let's talk about staining. So stains on a chemical level are actually considered salts that are composed of a positive and a negative ion. Just like sodium chloride is composed of the positive ion sodium and the negative ion chlorine put together to form table salt, stains are composed in the same way. However, in the case of a stain, either the positive or the negative ion is something called a chromophore, which is a chemical compound that has a color associated with it that's visible to our naked eyes. Thus, stains can be classified in one of two groups. Basic stains have chromophore as their positive ion, and acidic stains have a chromophore as their negative ion. And this has an impact on how the stain or dye is used. In basic stains, which have a colored positive component, the chromophore, being positive, will be attracted to negatively charged things. The opposite is true of acidic dyes, where the chromophore is a negative ion. Hence, it will be attracted to positively charged things. At a neutral pH of 7, the surface of bacterial cells are normally slightly negatively charged, which means that the chromophore in basic dyes, which are a positive charge, are attracted to them. We call this positive staining, and there's an image here on the left of what a positive stain using a basic dye would look like. Each of these filaments represent a bacterial cell to which a basic dye has been adhered when the positive charged chromophore was attracted to the negatively charged surface of the bacterial cell. In acidic dyes, the opposite is true. Because acidic dyes have a negatively charged chromophore, that colored component is actually repelled by the bacteria. It wants to attach to anything but the bacteria. So this creates a negative image, or as we call it, negative staining, where the background is colored but the bacteria appear uncolored here. So stains can be used in different ways to visualize bacterial cells and other types of microscopic organisms. The most straightforward way to apply a stain is called simple staining. Simple staining consists of using a single basic dye, not an acidic dye, but a basic dye, which is applied to highlight the organism's outline of their cell wall and or cell membrane, as well as their DNA. Not only is the cell wall / membrane negatively charged, the DNA of an organism is also negatively charged, and this means that the basic dyes will be attracted to the DNA as well. In this image right here, this is a simple stain of human cheek cells using the basic dye methylene blue. And not only can you see the outline of those cheek cells, you can also see that each cell has a little dot inside of it that's slightly more darkly colored than the rest of it. That little dot is the nucleus, the part of the cell where the DNA is contained. Simple staining is used to visualize the shape of cells, and in some cases it can be used to visualize internal structures, particularly those that contain DNA, like the nucleus. Beyond simple staining, a more complex way to apply stains is in something called a differential stain. A differential stain is exactly what it sounds like. It relies on multiple different colored dyes that can be used to distinguish or differentiate between different types of bacteria, hence the name differential stain. The most common types of differential stains that are used clinically today are called the Gram stain and the acid-fast stain. The image that you see right here is of a Gram stain, and as you can see, there is not just one but two different colors going on here, because the Gram stain, being a differential stain, allows you to distinguish between different types of cells in the same sample based upon a characteristic of their cell wall. So let's take a look at how the Gram stain and the acid-fast stain work, starting with the Gram stain. The Gram stain is a 4-step process that will be covered in one of our labs this semester. The first step involves the application of what is called the primary stain, the first of the two stains that is used in this differential process. Usually the primary stain in a Gram stain is a dye called crystal violet, which is applied to the smear, and it stains all of the cells in the smear purple regardless of cell wall character. In the second step, the primary stain is washed off, and then a mordant, which is the chemical compound iodine, is applied to amplify the color of the primary stain. The iodine drives the crystal violet further into the cell wall and causes the dye to be sort of glued to the cell wall in order to enhance the coloration. In the third step, a decolorizing agent, which is the chemical compound alcohol, is applied to the smear. And this decolorizing agent will cause the primary stain of the crystal violet to be removed and washed away from some species, but not others. So in other words, some species will retain the purple color and others will lose it, becoming colorless. In the fourth and final step, a secondary stain or counter stain, which in the Gram stain is safranin, is applied to color the species that lost their color during the step 3 decolorization. At the end, if you have a mixed sample, you can end up with some cells that have a purple color retained from the very first step of applying crystal violet, and some that have a pink color that came from the fourth step of counterstaining with safranin. Safranin is a pink reddish colored dye, which is named after the reddish colored spice saffron. So what's the point of that Gram stain series of steps? Well, the Gram stain allows you to distinguish between cells based upon whether or not they have a thin or thick layer of peptidoglycan in their cell wall. In chapter 1, we learned that bacterial cell walls are composed of a substance called peptidoglycan, but not all bacterial cell walls are the same in terms of their structure and the layers of peptidoglycan in terms of their thickness. Some bacteria have a thick layer of peptidoglycan in their cell wall, and these bacteria end up retaining the primary stain of crystal violet, meaning that they stay purple all throughout the stain. These bacteria are classified as Gram positive. So we call bacteria with a thick layer of peptidoglycan Gram positive. On the other hand, bacteria that have a thin layer of peptidoglycan in their cell wall will lose the primary stain of crystal violet during washing that's pulled out of that thin layer. They will then take up the counter stain of safranin and turn pink in color. And we call these types of bacteria gram-negative. And why is this all important? Well, it turns out that the thickness of the cell wall has consequences for what types of antibiotics are most effective against the bacterial species. Certain types of antibiotics work for gram-positive species, while others work better for gram-negative species. And we'll learn more about this in the future lectures, but this is the utility of the gram stain in a clinical setting. The acid-fast stain also has utility in the clinic. Acid-fast stains are used to identify bacteria that have waxy material in their cell walls. This particularly includes bacteria from the genera Mycobacterium and Nocardia. Mycobacterium is a genus that includes two important human pathogens, Mycobacterium tuberculosis, which is the cause of the respiratory disease tuberculosis, as well as Mycobacterium leprae, which is the cause of the nervous system disease leprosy. Nocardia are a genus of bacteria wherein some are pathogenic, but they are usually only seen in immunocompromised individuals. So the main differential purpose of acid-fast staining is focusing on the genus Mycobacteria. So being a differential stain, the acid-fast stain again consists of 4 steps. The primary stain in this case is not crystal violet like it was in the gram stain. Instead, it's a chemical compound called carbylfusion. It is applied to a fixed bacterial smear to color all of the cells pink. Then, the slide is gently heated over a beaker full of hot water, and the heat serves as the mordant. So in the gram stain, we saw that the chemical compound iodine served as the mordant, but in this case, it's the heat. So why would heat be an appropriate mordant in this case? Well, we are looking at bacteria, or rather, we're looking for bacteria that have waxy material in their cell walls. These are a special case of bacteria that, in addition to peptidoglycan, they have this lipid, fatty, waxy substance that covers the surface of their cells. And one thing that we know about wax is that with the application of heat, it melts. So in this case, the application of heat will allow the primary stain of carbylfusion to drive through the outer layer of cell wall, through that waxy substance, all the way into the cell membrane. After the mordant is applied, we once again decolorize. The slide is decolorized with acid alcohol, and this removes the primary stain of carbylfusion from the cells that do not have a waxy cell wall, causing them to become colorless. Finally, in the last step, a counter stain is applied, and this counter stain is usually methylene blue. It is taken up by cells that lost their color in step 3. That is, the cells that lack a waxy cell wall. At the end of it all, you would expect cells with a waxy cell wall, which we call acid-fast cells, because their color stands up in the presence of acid alcohol decolorization. These cells are pink in color, whereas non-acid fast cells, ones for which the color was pulled out of them, drawn out of them by the acid alcohol wash, these ones are now purplish blue in color because they took up the counter stain. The pink ones, the ones with the waxy cell walls, could potentially be Mycobacterium tuberculosis or Mycobacterium leprae. Now, in this checkpoint here, I want you to try your skills out interpreting an image of a gram stain. I've circled a cluster of bacteria here, and I want you to tell me if they are gram positive or gram negative based on their color. Likewise, in this checkpoint, please tell me in this acid-fast stain whether letter A or letter B could potentially be the bacterium Mycobacterium tuberculosis. So at this point, we have covered simple staining and we've covered differential staining. The last type of stain that we need to talk about is actually a group of different types of stains that are considered special stains. Special stains are used to identify specific structures within bacteria. So we'll talk about 3 examples of special stains, starting with a stain for capsules. Some microbes contain, in addition to their cell wall, a protective coating called a capsule. And it's important to know whether a bacterium has a capsule because it tells you something about its level of pathogenicity or virulence. Virulence is defined as the degree to which a pathogen can cause disease. Bacteria that have capsules have a greater ability to cause disease because the capsule, the protective layer, makes them more resistant to our immune systems. In addition, this layer called capsule does not take up most dyes and rather appears as an unstained halo around the cell when stained with a negative stain or an acidic dye, such as India ink or nigrosin. That's what you can see in this image right here. This is a negative stain of a bacterial sample which is clearly exhibiting capsules as a blank halo color around the outside, given that capsules resist colorization and look like a clear halo. So next we have endospore staining. Endospores are these highly resistant dormant structures that can arise in some genera of bacteria under stressful environmental conditions. I like to think about these as being like the bacterial bomb shelters. Under stressful conditions, some bacteria, particularly those belonging to the genus Bacillus or the genus Clostridium, are able to produce endospores which are highly resistant to drying, to high temperatures, to low nutrient conditions, and by storing their DNA in an endospore, these bacteria can wait out the stressful environmental conditions and then germinate later once conditions have improved. That's why I call them the bacterial bomb shelters. In order to detect the presence of endospores, a stain that uses malachite green applied in conjunction with heat as the mordant, then decolorized in water and counterstained with safranin can reveal whether endospores are present, because the malachite green will be taken up by any endospores in the sample, whereas the safranin will counterstain normal bacterial cells. And that's what you can see right here in this image. The last special stain that we'll talk about is the flagella stain. Flagella, as we mentioned before, are special appendages that many bacteria possess that are used for movement. They are these whip-like structures that hang off the ends of bacteria, and as they rotate, they can propel the bacteria around its environment. Normally, flagella are too fine and too small to be seen using a normal light microscope. However, they can be visualized using a special stain that applies carbofuscin and a mordant to coat the flagella and essentially increase its diameter until it is thick enough to be visualized under the microscope. As you can see right here in this particular bacterium, we have not one but multiple flagella that are present on either end of the cell. This leads us to checkpoint number 5. Now that we've discussed differential staining, I want you to take the steps of the differential stain and place them in the correct order from first to last. The mordant, the decolorizer, the counterstain, and the primary stain. Lastly, I want you to tell me whether the special type of staining that is used for identifying endospores would be classified as a simple stain or a differential stain. And tell me why as well. And once you finish this checkpoint, you are finished with chapter 2.